All right, so let's jump into the material. So the first thing is that uh, we are discussing this against the viruses. And remember that the vaccines are against many different pathogens, including a lot of different bacteria. But we will be doing this in context of the viruses. And as you can see, that life expectancy, the US, as per the CDC, right here, it's on a constant increase. But unfortunately, what happened that there is a little bit of dip after the 2012. I'm not going to talk about that, and that is related to the opioid crisis as such. And the vaccine mobilizes the host immune system to prevent the virus infection. And you had seen that from the previous chapter, that your adaptive immunity kicks in, and there are some memory B cells and the T cells that remain in your system lifelong most of the time. Sometimes it doesn't work, and we'll talk about that, why it doesn't work, and you have to take the vaccine again and again uh, after a couple of years. But that is basically the success of that vaccination, and it breaks the chain of transmission. You can see that there is a dip. This is just during the World War I as such, but you do not see that much of dip in the war during the World War II, because already at that time there was the antibiotics actually came out. All right. So the next thing, what we are going to see, that this is the basis of vaccination, and this is how this is background information from your previous chapter of the immune system, adaptive immune system, that you just first get the infection and you will have an initial immune response. And this immune response will include your innate response as well as adaptive response. Your initially antigen presenting cells going to present the antigen to the T helper cells. T helper cells will mobilize the cytotoxic T cells, the B cells, the whole yard. And after that, the infection will be cleared and you have a protective immunity. The memory B cells and the T cells will reside in your lymph node, and they will stay supposedly for the rest of your life, if not a considerable amount of time. And then what happened, that if you just get another time after that, if you have a healthy repertoire of T and the B cells, you get a spike in your initial immune response. But at this time, what you're going to see, that mild or no inapparent reinfection. You are not going to suffer from that. Before the virus can actually do anything, your immune system can kick in much faster than the first time. You can see that's a sharp rise because you will already have the B cells and the T cells, which can recognize that pathogen. And this happens very fast. It does not have to wait until, see, that 5 to 7 days. It's just within a 1 or 2 days before the virus can do any harm. You will have a sharp increase in the immunological immune reaction, and that will contain the disease. This is basically the basis of vaccination. So before we go into the different types of vaccine, one of the important thing that we talked about in the previous chapter as well, that what is an active and passive vaccination. So the active is instilling into the recipient the modified form of the pathogen, here virus or material derived from that, to induce the immunity to the disease. And this is the basis of most of the vaccination, or active vaccination. When you get the infection and get immune, that is your active vaccination. Your own immune system is producing the agents that is required for the protection in the future. That is your active immunization. Passive immunization, on the other hand, instilling the products of the immune response, which are antibodies or immune cells like cytotoxic T cells, and you infuse or inject them to the patient. Very different. Here you are injecting a virus or the pathogen, say for a vaccine. Here you are not injecting the virus or the pathogen. Here you are injecting the patient with an antibody or immune cell that can recognize the pathogen. And this is an emergency purpose short-term protection. Classic example similar to this is when you get a snake bite. Basically, the shot that you get is a collection of immunoglobulin G, which will immediately go and bind to these phosphatases, which actually do the harm in a snake bite condition. And that will contain the harm that can be caused by those poisons. So that's passive immunization. The same way, and the interesting that what happened that you all know that the Regeneron antibody that was used on President Trump as a clinical trial, that was a passive immunization. He received the antibodies, and that immediately neutralizes the viruses. Remember that if they are monoclonal antibodies, this is a much more expensive way, and sometimes may not be feasible for the mass eradication of a disease. And in the SARS-CoV-2, this cannot be used for the billions of people as such. So we have to wait on this active immunity. So let's see that what is a natural passive vaccination. This is the second thing. I'm sorry. The second thing that we are talking about. Natural passive immunization comes from the mother to infant, and this actually happens in utero. So if the mother is immunized against a pathogen, like have taken vaccines or exposed to a pathogen, the mother's antibodies can cross the placenta and just get into the infant's circulation. And then even after the birth, just immediately after the birth, before the milk glands are active, it takes about a day or two, there is a colostrum which actually comes out of the mammary gland that was extremely rich in these antibodies, and that protect the newborn child. And what happens that you can see right here, the birth. At birth, pretty much the amount of antibody that the newborn actually has is almost to the adult value of that. So at the highest level. And the reason for that, why it is that our immune system takes a time, almost an year, to fully develop. So you can easily see that this is the IgM, IgG, immunoglobulin G, and IgA secretory antibodies. These are the pentameric antibodies, and these are the immunoglobulin G is the main type of antibodies, which is the most prevalent one. Takes a little time to develop. And in that time, the mother's antibody actually protects the child. And one of the interesting things here, you note that usually the first shots or immunization or the vaccination are not administered to the newborns before usually 6 months of age. Usually you get at 6 months, then 12 months, 18 months, if they're repeated shots, but usually you don't get a vaccination before 6 months. And see that what is the reason. If you give a shot to a child when it is 3 months or 2 months old, at that time, you have a large amount of mother's antibodies circulating in that infant. And what will happen that these antibodies will recognize that the pathogen that is going inside that child as a foreign source, and it will inactivate that immediately, and that will not elicit an immune response, and that will not go into the immunological memory of the child. So once the mother's antibody is waned off, about say 6 months around that time, you get the first dose, but you get a booster dose after about 12 months or 12. But the thing is that this is the natural passive vaccination from mother to infant, protects the child at the initial stage when our immune system is not that very well developed. All right, so after that, we are looking at a complicated slide, but let's take a look one at a time, that how we do make these chimeric antibodies. And there is an advancement of these, and that is being used with many different ones, like in Ebola passive antibodies, then SARS-CoV-2 passive antibodies. And let's take a look at how this actually works. So just a complicated slide, but we are just concentrating first on the very left side right here. So if we just look at the antibody, the typical structure, you can see that it has 4 chains. So these 4 chains, if you just look at that, this right here, this one outside, this is the L chain or the light chain. And then this one inside, which is joined right here, it goes all the way down, is the heavy chain. This is a heavy chain, and this is the light chain. Now, what you have, if you carefully look at that the tip of these chains right here is a variable light, variable heavy, variable light and heavy. And after that, you have a constant heavy, constant light, and then you have a constant heavy and a constant heavy region 3. So all the constant regions are same in a typical isotype of an antibody. So if you take an IgG of the human, your constants region CH1, CHL, CH2, CH3, they are all the same in all IgG. But what will vary is the variable heavy chain and the variable light chain, because these are the recognition for the viral antigen. And we call this as a CDR, or complementarity determining region. Basically, these are like the molds which will fit into one form of the virus or the pathogen. So to do this, how it is, you can see that heavy chain has at the tip variable heavy, then constant 1, 2, 3 region, where the light chain has the variable light and the constant light. So you have the constant region and the variable region. So here, suppose this is a mouse antibody, and we raise this antibody against the virus or the pathogen. If you take this recognizes the viral pathogen, but if you take this antibody and inject it to the human, it will not work, because all these regions of the constant regions are of mouse type, not human type. High immunogenicity, and immediately will attacked by, this will be recognized as a foreign molecule and immediately attacked by the antibodies of the humans, and the whole of the antibody will get neutralized. It will have no function as a passive immunity. So what you do? On the other hand, this one in the yellow is the same thing, which is the human antibody, pure human antibody against the virus and antigen. And if there is no immunogenicity as such, because it is your own antibody, human antibody, the variable and the light chains are the same. So what you do in the chimeric antibody, you take the backbone or the constant region, see that this is the constant region of the human antibody, and you only change the variable light and the variable heavy region with the mouse antibody. And this is the grafting of that. And this will have a much less immune, this will be able to bind to the virus or the pathogen in the human system, but this will not be immunogenic because the rest of the antibody is from the human being. So this is your chimeric antibodies. So let's take a look at a different perspective. So here we are looking at a little bit of different ways that even we just go a little bit farther. So here you have a mouse chimeric antibody, and these are all produced initially in the mouse, and then they are made into the hybridomas. And this is the human antibody, and they are made into the transgenic mouse human hybridomas. This is kind of funny. They have typing errors like me as well. So what you do that you take these tip of this regions, you take this one off, and then you replace those with these variable heavy and the light chain. You can easily see that. This is the chimeric antibody that was shown in the previous slide. Now we have even made more progress in the antibody technology by the genetic engineering. We specifically change some of the key residues which can which do have the complementarity of the site or the viral site, but they also, their scaffolding areas are from the human being. So this chimeric or humanized antibody does even better than chimeric antibodies, because they will not react and less immunogenic to the humans. They will only detect the virus. And this is the latest antibody genetic engineering. And we will talk about detail that how we develop that in a special topic. And this is done by complete genetic engineering, which is the variable gene cloning. And onto that, you put that CDR grafting. The grafts are in yellow, and the CDR is complementarity determining region. You only graft the amino acids, and accordingly their nucleotide region of those amino acids, which are responsible for determining the antigen. And you have to screen a large number of antibodies to do that and find these common region which can recognize the viral pathogen, and you graft into the scaffold of the human being. It's an amazing way that how you can immunize the antibody. The Courtney in the covered this Regeneron's, and we'll come back to that again. And this was the type of antibody that was used in President Trump with the Regeneron produced humanized antibody. All right, so this is, on the other hand, is a three-dimensional structure of these antibodies, just in different perspectives. So if you carefully look at that this is right here is the mouse antibody, and right here is the human antibody. And if you very carefully look at that you can see that this is the heavy chain, right here is the heavy chain, and right here is a light chain, and see that this is the variable heavy, variable heavy, variable light in the human beings. And here it is not in two different colors. When you have the chimeric antibody, what you can see that your variable light, variable heavy chain, both of them have been changed, and this is the epitope determining region, and the rest is from the human being. But what you are seeing right here in the humanized antibody, as I said just before, this is the murine CDR sequences, which is called the CDR grafting, complementarity determining region grafting. Only the specific amino acids which recognize as the pathogen or the virus that has been grafted, rest everything is from the human antibody. And this is basically called the humanized antibody. And you can see that this is the immunogenicity. What it means that when you inject this chimeric antibody, you will still get some immune response, that these antibody will be attacked by your own antibodies, so that they will not be 100% in neutralizing the viral antigen. As they will be less attacked by your own antibodies, their amount will be much more in your circulation to attack or specifically bind to the virus, and not neutralized because it sees the mouse antibody. Because remember that any of the red that it is your antibody sees, it will be considered as foreign, and it will try to bind in there. And you can see that the red region is drastically minimized by this CDR grafting technology. So here is the passive immunization is a classic example, that the ZMapp of the Ebola virus. And you can see that there are 3 monoclonal antibodies binding to the receptor binding region. And this was a chimeric antibody, and it was called a ZMapp. And this is now being produced as a recombinant in the tobacco plants. And this actually helped the 3 personnel who actually contracted Ebola from Africa, and then they were flown into the United States in a C-17 or something like that, these planes in a quarantined condition, and they were given this passive immunization, and all of them actually did survive. So this is also the passive immunization for Ebola. On the other hand, this is the Regeneron's humanized antibody for the SARS-CoV-2. What you are seeing in this, that this is the viral receptor binding site, or called the RBD. And you are seeing that there are 2 portions, just only you are seeing only the portions of these Regeneron 2 antibodies. These are their names. But one of the antibody binds right here, another antibody binds in here. So what you are seeing that in the RBD region, there are 2 epitopes. The epitope is just the amino acid sequence which is recognized this antibody. So this is your epitope 1, for example, say. Here, these amino acid sequence are the epitope 2 recognized by this monoclonal antibody. Remember that these are all monoclonal antibody. Each one of them will recognize only this. So advantage of that if you just use a cocktail or 2 of them, you have the chances of them binding or neutralizing even more, because you are binding to this region, you are binding to this region, and that is much more effective than just using just one antibody. This is the same concept that in Ebola virus RBD, you can see that there are 3 monoclonal antibodies binding to 3 epitope binding region, and that's how it can replace the receptor binding region of the RBD. So think about that. This is the receptor that you have right here, which are the glycoproteins that you are seeing right here. Those are on the kind of like so this is, think about that. This is the Ebola virus envelope, and this is the glycoprotein sticking out. And these are the 3 regions where these monoclonal antibodies are binding, which is basically this. And this RBD binding, remember that this viral receptor binding, this RBD binding region is from the spike protein of the SARS-CoV-2. All right. Now, that's active or passive immunity. Now we are going into the real deal, that how we actually give the vaccines, which is the mainstream vaccines, and which elicit the active immunity. So here you have the virus structure, and we're going to see that there are inactivated vaccine, attenuated, recombinant vaccines, DNA, mRNA vaccines, and subunit vaccines. So we are going to talk about these, but we are not going to talk about the toxoid, carbohydrate vaccine, and conjugated vaccine. The reason that these are mainly implicated in bacterial type of vaccine, we usually do not use these technologies for the viral vaccines. That's why we will just concentrate on this, basically. So let's take a look at one at a time. A lot of content. So the first thing where it all started is, you all know that it was Jenner. Edward Jenner vaccinated Peter because he got that observation that the milkmaids get small pustules on their skin from the cowpox, and which is not very it doesn't cause much disease in the human beings. But what he observed that these milkmaids, they don't get smallpox, and so he took out a pustule and just infected this Peter with that. This was the first vaccination, and at this time he will probably end up in the jail. But again, at that time it was possible. And then he challenged Peter with the smallpox virus for about 2 weeks. And at that time, they had no idea about the virus and how long it takes the immune system to work in. And the good thing is that he waited for about 2 weeks just by chance. If he have injected the smallpox within a couple of days or a week, there will be no adaptive immunity from that cowpox vaccine that he gave, and the young boy could died of smallpox without any immune protection. But it was just by chance that actually happened, and that started all these rays. And this is the Kensington Royal Garden. If you just anybody been to London? So this is just across. This Royal Garden is just across from the Buckingham Palace. And if you go to London, you will be seeing that immortalized Jenner in London. And this is very heart of London. So that's where it all started. All right. So after that, I know that it is a little bit disturbing, these images. So if you don't like to, you can just look away from the screen, and you can delete those if you feel uncomfortable from your PowerPoint as well. So how the smallpox was initially given, that a bifurcated needle where you just take a little bit of these varicella or the variola, not varicella. Varicella zoster is for the chickenpox. The variola virus that is given by this bifurcated needle. And the reason is that it actually replicates the best just in this subdermal region, subcutaneous region, and you will get at the primary vaccination site reaction. And after 1980, if you are born, you have not received any vaccination. That I do have a vaccine. I do have a scar in there as well. And again, remember that this is 1 centimeter, although it looks kind of to today's extent that is gross, but it was nothing. I mean, like you just within 3 weeks, it will just dry up and you are fine. But just thinking about that this is the reaction, and you didn't want to have this with a child of smallpox. And you can easily see that it disfigured, made people blind, made people, it was, I mean, like it killed probably more people on earth than any other disease. But it is eradicated. So you can easily imagine that what is the power of this vaccination. But even after 2 centuries, the fear of the vaccination still remains. This is an 1802 caricature of Jenner vaccinating patients, and they are developing cow-like appendages. So you can just imagine that still you have a very vocal anti-vax group who still are not convinced about the vaccine still after 200 years. I'm not going into the political thing that beyond the scope of this class anyway. So these are some of the timelines that you have seen, and these are the ones that I was talking about that I'm not going to go into the detail of that. This is just for your understanding that where is what happened. and I will just save some time on these. So again, rabies followed by Pasteur, yellow fever, then polio came out. Just an important thing that Jonas Salk is discovered the polio vaccine. This was an inactivated vaccine, and we're going to talk about that. Injected 4 shots, whereas the polio vaccine oral, that was done by the Albert Sabin, which actually eradicated the polio. Jonas Salk's vaccine actually could not, and we'll talk about that, why it is so. So you can easily see that large-scale vaccination campaigns can be successful, and this was done by the World Health Organization, and it was 1980 in May. They declared that the planet is not the planet is free of smallpox, but not rabies, not yet. You have some countries where you can see that. But you can see that this is the power of the vaccine, that the inactivated vaccine, Jonas Salk's vaccine, it dropped dramatically, but could not eradicate. The oral vaccine of Albert Sabin actually came out, which is a trivalent with 3 different strains. And finally, it is of a decline. Measles vaccine, you can see that large-scale vaccination is still there, but measles is still a problem in many of the developing countries. But you can see that just within 2000 and 2012, 13.8 million people were saved by these polio vaccination. So large-scale vaccine campaigns can be very, very successful. And the next one is that how the key concept actually works is the herd immunity. You see, listen to that in the media every time. So basically, the concept is that a population scale immunity. So what happens that when a good number of people are infected and a good number of people are susceptible, they don't have an immunity, it is very easy for the virus to spread, which we are seeing with the SARS-CoV-2. On the other hand, if you actually do see that a good number of people are immune to the disease, even if there was one person, there are much less susceptible people and the disease transmission is blocked. And that is the herd immunity, and that works. So this means that 100% of the population doesn't need to be immunized or vaccinated. And if you have a critical threshold of herd immunity is reached, you will not get the disease. And how this is calculated is very, very easy. So R naught or R0, R naught determines the percentage of the immune population to reach the herd immunity. And how R naught is calculated? R naught is if you have a particular virus, how much of the virus is from one person, how many of the person can get infected. Say, for an example, roughly, I'm just saying, for measles, it's very high. It's about 20. So what you do that the calculation is 1 minus 1 over R naught. And the fraction that you get, that is the percentage that you have to, and this is, I put that in the notes, very easy. That number is the percentage of people need to be vaccinated before it can be used or the herd immunity can be achieved. So if you calculate for plugging the number 20 for measles, you will see that it will be about 95% of the population need to be immune. On the other hand, if you just say for SARS-CoV-2 is in between 2 and 4 people, or the R naught is about 2 and 4. So if its R naught is about 4, then if you just calculate, you will see that about 75%, 1 minus 1 over R naught, which is 4, 1/4. And that will be about 75%, 1 minus 1/4, 75%. Very easy calculation. So 75% of the population need to be immunized before you receive or achieve the herd immunity. Sometimes that number is even lower where you do not see. Say, for example, measles, instead of 95%, which is the textbook value or calculated value, probably about 70, 80% if of the population, if it is immunized, still it works pretty well. But there are some of the misconceptions that goes around. And this is what it is. Reaching the herd immunity but natural infection is too much cost to pay, particularly if it is a lethal virus, for an example, SARS-CoV-2. Yeah, I mean, like it's funny, but this was a recent cartoon that herd immunity achieved, but it is before that, too many people will die. And the only way that you reach herd immunity is by vaccination, not by natural immunization. So, again, I'm not going to the political detail commentaries on that, but you can just well understand that it is not, it is very dangerous, and in a lethal condition, you just cannot achieve that. You can take those risks to some extent in particularly virus which can cause much less amount of invasion or something like that. But not with the ones which have high lethality, particularly if you have a disease condition. All right. So on the other hand, what are the other requirements of these vaccine that you can easily see? That you have to not only just give you a proper immune response, it does have to give you a proper T helper response and T helper 2 response. See, the T helper 1 response actually calls the CTL, or the cytotoxic T cell mediated immunity, whereas the Th2 is the antibody response. Usually what happens that if you have an acute virus, which comes in, goes out within 7-10 days, you want the response to be skewed towards this Th2 response. You can see right here. Whereas what happened if you have a persistent virus, like an herpes virus, which stays inside the cell, don't come out, antibody, if the virus is not coming out into the interstitial fluid or the blood, this antibody response doesn't work. You have to have a cell-mediated immunity, or a cytotoxic T lymphocyte, which can recognize an infected cell and kill that. So, for example, a persistent infection where the virus is inside a cell, a more dominant Th1 response is responsible compared to an acute infection. So this balance is very important. So it is just not that a vaccine is great because it just elicits an immune response. And these are some of the steps which are required for the success of a vaccine. All right. So after that, the effective vaccine distribution and storage, these are some of the issues that you have, and induced protection and immunity in the population must be long lasting and low cost. And some of the challenges that what you have, that in many of the developing countries, the vaccines, particularly this actually happened with this smallpox and the polios, that there is no electricity even. So this is a thermos which can maintain the cold temperature, and it can be run by the gas or the generators which are used with this, and that can keep cold these vaccines for almost over a month. And these are some of the things that has been developed. This is a commentary that I'm just putting it out there. You all know that the Pfizer's actually came out with this mRNA vaccine, but they declared that the 95% efficacy. But again, we'll talk about that what does that mean? But they need to store that vaccine at minus 80 degrees Celsius. And think about that. These minus 80 degrees Celsius are in some of these labs that you see in the biological sciences, other biochemistry, and all different departments that you actually see. And these minus 80 degrees centigrade freezer actually costs 10K and above. It is not minus 20, which is your refrigeration or freezer in your household. This is minus 80, ultra low. And you can well imagine that how many, even in the developed countries, a rural area may not even have a minus 80. So this could be a problem, and that has to be solved before this vaccine can be available to mass. All right, so here is the viral vaccines which are licensed in the US. This is just for your information. Again, remember that you do not have to memorize any of those. And here, just one thing that I'm just pointing out here, that there are many engineered vaccines which are in the engineered on a virus. And here it was engineered on a vesicular stomatitis virus, which is an animal pathogen, but not a human pathogen. And Ebola virus glycoproteins are replaced on those. And this RVSV-ZBOV is licensed in 2019. This is the first recombinant viral vector vaccine. So there is just only one in the market. They started developing this in 2008 and 2009 took them 10 years before it could get licensed. There are about 11 or 12 adenovirus vector vaccine for the veterinary use, but there is none for the human use. But there are several ones which are under the development, but we'll see that how fast they can get licensed. All right, so after that, we are going into the science of an art of vaccine making, and we just put the word art because it is really an art. It is just not the science, because that sometimes you do everything right and there are so many things that can go wrong that a vaccine may fail in that, and that's the reason that we call this that one. So we will going to talk about that. What is an attenuation, inactivation, fractionation? We talked about this virus vector vaccine, DNA vaccine, an expression protein vaccine into nanoparticle, and as well as we're going to talk about the mRNA vaccine. So let's take a look at one at a time. So the first, before we go into the vaccine, one thing is important here, that when we inject this vaccine, except a few of them, remember that in the DNA vaccine and those, this is not required. You need something which is called an adjuvant, except DNA RNA vaccines. So what the adjuvant is, this stimulate early process in the immune recognition, which is basically to agitate your site of injection. So it means that it will create an inflammatory response so that more of your antigen presenting cells come in there, cause an inflammatory response, and more of your immune system is activated. And this is the reason that the shots are sometimes painful. Slow release of the antigen is a site of inoculation and cause inflammation. What are these? Say, for example, aluminum hydroxide, squalene, phospholipid A. And see that this monophospholipid A, this is basically from a bacteria. But what happens that it will not cause any harm or anything like that. But when you inject these things, or say aluminum hydroxide, this will irritate the region which is the injection site, and that irritation will cause the inflammation, which will attract more immune cell so that a good robust immune system can be achieved. And so this is what is called the adjuvants, and these are the ones which are usually used. The vaccines are mixed with these. All right, so the first thing we are looking at is the inactivated vaccine. And how do you do this? You just take these virulent parental virus and you just use formalin or a beta propionlactone or non-ionic detergents, and you have an inactivated vaccine. So you basically kill the virus in that process, that it will not be able to replicate, but your antigenicity is not compromised. And that is basically what we called as an inactivated or killed vaccine. And still, as I've said, that still this is the classic technology and old and the gold standard in that process. So let's take a look at some of the historical perspective. This is the polio, which caused the poliomyelitis, and see that how debilitating disease this was. These were the iron lungs and the ventilator. And the reason that they are in there, because they are paralyzed, that they cannot breathe on their own. And many of these patients actually stayed in these ventilators or the iron lungs for the rest of their life. And very different in that. And this was the condition of the poliomyelitis. And so basically what happened, that Jonas Salk in 1955 first got the vaccine for the polio virus. That's what you're seeing right here. And for the clinical trial, actually, Roosevelt, FDR, actually started this March of Dimes, and that funded the Jonas Salk polio vaccine. Remember that it was not also, you may not even know that it was not portrayed much, but the FDR had a polio. And this is one of the rare pictures where he's being helped to get out of this car as such. And he started this March of the Dimes that you still see. So here, some of the commentaries about the polio virus. You can see that 1954 National Foundation for Infantile Paralysis. This is basically the March of the Dimes. About 1.8 million people were given these vaccines, greater than 50% protection. But there was one problem, and see that this is what happens when you have, even it's inactivated vaccine, but there was a problem. So there was a family-based company called Cutter Company, and this was in California, and they actually could not inactivate the vaccines, and that has caused 40,000 cases of polio to the vaccinated children, with 200 actually got kind of varying degree of paralysis, and probably 10 died out of that. So you do not want those things to be rushed. And even with the inactivated vaccine, you need a great quality control. And that's one of the problem that you have. So here, the Salk vaccine actually did not stop the vaccination of polio, because the problem is that, remember that the virus gets through the intestinal tract and it goes from the intestinal mucosal surface to the lymph node, and only it reaches the brain through the blood. But that initial part that it will get into the interstitial area, and just just the shot into the arm it did not help the vaccine to get into the lymph node from the mucosal surface. And that's the reason that what we are going to see, that the oral vaccination of Sabin actually was required to get the polio in control. Next, we are looking at the influenza virus, 3 different types, and these are just, we have to skip those very quickly. So you can see that right here, large number of people die due to that, and protection correlates with the serum hemagglutinin and neuraminidase. These are some of the antibodies, and you can see that they were all produced in chicken eggs, basically, but now most of them are done in cell cultures. And you can see that what they do, that there is a reassortment with most of these RNA segment happens. You can know that these HA segments all reassort, like it could be H1N1, it could be H2N2, H5N1. All these different things happen, and that's the reason that you have a multiple vaccine like H1N1, H3N2, and this is a 2019-20 vaccine. So this is, if you get a shot this year, you will get these strains, and this is circulating mainly. So how this is determined? So this is the timeline. So what is happening, that before you get the vaccine, say for example, for next year's vaccine, the surveillance will start in January of 2021 to get the vaccine for 2021-22. And you can see that you select the strain for first few months and then standardize the antigen and formulate test package, and it is available from approximately September. What happened in 2014-15, the selected strain mutated right here around April, May, June, and the vaccine was not that effective. So this can actually happen, and this is the reality of that. So this is how the influenza vaccine, and there is no universal vaccine, but there are many research that is underway to make an universal vaccine. The next one is the attenuated vaccine, where the viral replication occurs, stimulate immune response, but the infection induces mild or no apparent disease. So how this is done, that usually the parental virus is passaged many times in cell culture or in a non-permissive or slightly permissive animal, like it's a human virus passage or in the monkeys many times, and somehow you get an attenuated strain which has lost the ability to surmount a disease, and that is used as an attenuated strain. And how it happens, that you can easily see the difference in between that. If you have an inactivated virus vaccine, you have an initial dose, just a little bit of response. You get a second dose, a booster, and then you get more and more response. So you need many more boosters to get to the level of the inactive. On the other hand, if you have a replication competent virus vaccine, like an attenuated vaccine, you can see that as the attenuated virus does have all these surface antigens, you will get a robust, and it could be just amplification of the injected dose. You can easily see that, or immune response. So that's a replication competent virus vaccine. That could be either oral or nasal, or sometimes it could be injected as well. So here is how it is done. It is completely empirically derived. So you can easily see that this is a pathogenic virus isolated from a patient, grown in human culture cells, and then infect monkey cells. and the virus occurs some mutation that allowed to grow well in the monkey cells, but now these viruses will not grow properly in the human cells and may be a candidate for the vaccine. And this is completely a miracle, and you have to just screen thousands of culture transfers and all those kind of things. Extremely strenuous process. All right, so here is a flu mist, which is given to mainly the younger ones, and cold adaptive, temperature sensitive, attenuated in a ferret model. So basically, the human virus was given to the ferret, where it mutated and replicate only in the nasopharynx, produced a protective immunity. And this is the flu mist, which is also very, very effective. All right, so here is the Sabin's oral vaccine that you can see that we talked about. This was the attenuated vaccine and contained 3 strains, and this was given orally, and that also helped the mucosal surface as well, along and gave better protection than the inactivated vaccine of Jonas Salk. But there are some that we talked about. There are some mutation where the reversion of this polio vaccine does happen, and some people do get polio, and that number is out there. And it is a balance of that how much, I mean, like in 1 million people, probably 10 people can get polio. But so that's a concern. That's always a concern. But you have to weigh that the chances of having a debilitating disease and taking a chances. There is a fine balance between that. All right. So here is that how the attenuation of the poliovirus neurovirulence was done. And again, you do not have to remember or understand anything, but just how it is that you have these 3 different strains, and for 3 different strains, how these Sabin type 1, 2, and 3. These are the 3 Sabin vaccine strains that is a trivalent vaccine, and see 43 passages in kidney cell, 24 passages in the monkey, 43 passages in kidney cells, 5 passages in monkey kidney cells. And after that, you have 3 rounds of plaque purification. Here, they have to involve even a shrimp. You can easily see that. So depends upon that what you have, and this is what is we call as empirically derived vaccines, which with reduced neurovirulence, and ultimately you get the attenuated vaccines. All right, so after that we are looking at the subunit vaccine. So how you make that subunit vaccine? You can take the virulent parental virus and you can break it up where you have this fractionating individual proteins, say for example, and you can purify this subunit and inject. Or you can use a bacteria or yeast and clone the sequences of these antigens of the virus, which can recombinant technology produce the proteins or the virus-like particles, which you can inject. And that is basically called the subunit vaccine. You have, and we're going to see that what are some of these examples of this subunit vaccine. And the subunit vaccines are much safer than attenuated vaccines, as well as the inactivated vaccine, because none of these actually do contain any of the live viruses or even heat-killed viruses as such, but much more difficult, and they have poor antigenicity compared to the natural attenuated or inactivated vaccines. So here you have the recombinant zoster vaccine, which is called the Shingrix. So you have the enveloped glycoprotein, and this is a recombinant glycoprotein E is produced in mammalian cells, which are secreted of that and then adjuvanted with AS01. And this is a very, very effective vaccine for the shingles, which is the herpes virus. So this is a subunit vaccine, very, very effective though. Another hepatitis B virus vaccine. This is also another vaccine, and this is very interesting that how it actually happened. So the hepatitis B virus, this HBs AG protein right here, is produced in the yeast itself as a subunit vaccine. And you can see that these are very effective as well. So these are the incomplete virus particles, which does have all these surface proteins onto them and can confer antigenicity as well. So these proteins, when produced, they actually do have the hepatitis B virus antigens proteins are produced in the yeast, and they assemble into these empty virus particles, virus-like particles. It doesn't have the DNA or anything like that. This is a hepatinavirus, remember that, hepatitis B virus. And this can elicit the response in the patients. We do not have anything. We have hepatitis A and B, but the hepatitis C's clinical trial, there is nothing out there in the market yet. So it is not an easy job for every virus even. So here, human papillomavirus, remember that it is a cancer-causing virus, HPVs. So it's a cancer vaccine as well. And here is some of the details of these vaccines. And you can easily see that some men and women are both equally susceptible to that. So everybody should take this vaccine. But the thing is that remember that the developing countries cannot actually get these, because still the cost of this vaccine is probably around, I'm not sure, about $270 or something like that. So it's expensive. And that's one of the reasons that many of the developing countries actually doesn't even have that. But this is also a vaccine, which is a very, very important vaccine, and everybody should take this. On the other hand, this is the detail of that, how they are produced. The different companies like Merck, GlaxoSmithKline, these are the names, and these are the different types that are produced. And you can easily see that these are produced in the VLPs, or the virus-like particles. So you have the HPV, and L1 is the major capsid protein. What they do, that they clone these L1 into the yeast or baculovirus, which is a vector virus, and they produce, just like this empty virus particle, like these, they produce these virus-like particles which are empty. They don't have any HPV DNA inside that. And when you inject those, they will elicit the immune response. And very, very effective vaccine as well. So just a little idea that this technology, the subunit vaccines, are underway for the future influenza vaccine. The synthesis of the HA alone in cells leads to production of immunogenic particles. This can be produced in recombinant tobacco plants. And this has already been under clinical trial as such. And you can easily see that per square meter of plant can produce 20,000 doses, and the cost is less than 20 cents a dose. And this is a tobacco plant, and you can just develop these in the tobacco plants. Recombinant plants are making these antibodies. You just have to purify them. That's it. All right, so after that, we are going back to the attenuated vaccine, but from that, we will just see that why we're going back to the attenuated vaccine, because we are just going to see the recombinant vector vaccine from this backbone. So the yellow fever vaccine is available. It was produced in 1938 by, see that, 176 passages of a virulent wild-type CB strain, the chick embryo tissues. And as I said, that how many different types of techniques you have to do just empirically find that magic virus which is attenuated in that, and that was what done. And this is 500 million doses distributed, safe, effective. Remember that this is a Flaviviridae family. It's a plus-strand RNA virus, and the close viruses are the dengue virus and Zika virus, which are much more important now. Yellow fever is almost unheard of. You have to take it, although if you're going to certain parts of the world, that it's a travel vaccine, but you do not have to be immunized with the yellow fever vaccine. But dengue, Zika is also another. We have a dengue vaccine, but Zika is under clinical trial. So let's take a look at how it was developed based on the yellow fever vaccine. All right. So here what you are seeing, that this is a yellow fever backbone, or yellow fever, the plus-strand RNA, as I said. So you can see that UTRs, and then these are all the proteins. And remember that just like a polio virus, these are all the polyproteins which are cleaved into the proteases into the individual proteins. And what you are seeing right here, this is the envelope protein, glycoprotein right here. This is the E. So to develop the dengue vaccine, they took this CB strain, this one, which is the attenuated strain we used for the yellow fever vaccine. They took that strain, so all the sequences are from that strain, and they replaced this envelope protein with the dengue virus envelope protein. And that was the vaccine, which is the recombinant viral vector vaccine. So see that from attenuated vaccine, we got the base of a recombinant viral vector vaccine, and this is how it was engineered. And this is the Denvaxia. It's licensed in Mexico, Brazil, and Philippines. And this does not produce any protection on the dengue virus too. But sometimes remember that this vaccine does have a problem that we talked about, that this can cause this antibody dependent enhancement of the disease if the patient is already exposed beforehand to other type of dengue. That is a problem, and that is why it is not licensed in the United States and many other countries, because in Philippines there was a severe reaction that happened to 800 school children, and that's the reason that this is in some of the countries it is licensed, but in some of the countries it is not. So it is still not perfect. Now, this one is in the almost perfect condition, and this was developed by the based on the NIH, National Institute of Health, called the Tetravax DV TV003. And this is a tetravalent, means that it contains four different types of dengue virus attenuated virus strain. And how these attenuated strain actually work, most of them actually got attenuated by the deletion of a 30 nucleotide deletion from the 3' UTR. And if you just remember to go back, that we talked about that even in hepatitis C, that these regions can produce some of these microRNA based invasion of the cells. And that may be one of the reasons that how it is attenuated, that if you just take these untranslated regions out, it can get attenuated. And one dose, 100% protection to challenge. And this is the latest dengue vaccine that will probably, hopefully, be very successful in the developing countries where it is prevalent. So the next thing that came out in just a couple of years back, mainly in 2015, is the Zika. And you can easily see that in adults, it actually caused that Guillain-Barré syndrome. On the other hand, it caused acute myelitis, encephalopathy, meningoencephalopathies. But particularly in the child, the main problem that what happened is the microcephaly. You can see that this is basically the severe microcephaly, which can cause by the Zika virus infection, particularly if pregnant women are infected with that. So all vaccine making strategies are being tested. None is licensed yet. So you can see that there is a race for that, but still not licensed even after 5 years. And I think that the intensity of the disease also drive that how fast something can be done. So how this was, what are the different processes that is happening? So this is the same process that we talked about that replication recombinant virus vector vaccine used for this. And what was used in this process? So in this process, remember that there is a little bit difference in between that. See that right here, this was just even a dengue virus used a yellow fever virus, which is the same Flaviviridae family. Here we are doing an entirely chimeric virus. So what we are doing in the Zika virus, which is also Flaviviridae, and right here is the VSV, vesicular stomatitis virus. And these are the VSV protein, and the G is the glycoprotein, which is on the surface, which binds to the receptor. And as you can understand that as this is on the surface, this will be most immunogenic. And due to that, this glycoprotein is replaced by this membrane envelope protein, which is the E right here. E protein is replaced in a VSV. And this recombinant VSV was injected as the vaccine. And that is under the clinical trial. And this is the recombinant VSV. We call this was as a pseudo-typed VSV. Again, repeating, pseudo-typed VSV with the Zika virus envelope protein. And VSV is an animal virus. It is not a pathogen for the humans and doesn't cause a high immunogenicity against this, but it will not cause any disease. All right. After that, we have last part is the SARS-CoV-2, and it is a little bit intimidating in here. So here, what you can see that these are all the different vaccine candidates that you have. And these are the DNA vaccines, mRNA vaccine, nanoparticle based, even just we talked about VSA based, Merck is doing that. These are adenovirus based, and these are inactivated whole killed virus. These are being done by these companies. So all these names are the different companies that are doing that. So you can easily see that what is in here, the mRNA, Moderna, and Pfizer BioNTech, they are leading the race. Behind them are the Oxford, AstraZeneca, CanSino, Johnson & Johnson. They are in there. Novavax is also doing this. So these are all in this vaccine race. So let's take a look at how these actually do work. So the first we are talking about that is done, particularly this Oxford, CanSino, Johnson & Johnson doing that, and the Sputnik 5 in Russia. These are all adenovirus vector based. So there are two ways that you can do this. So you take an adenovirus, and then in the adenovirus, this is an attenuated strain of adenovirus, which does not cause any problem in the human beings. And you take this particular protein, which is the E1 protein. E1 is a transcription factor, you know that. So you take this E1 protein region and you put the spike protein, say for example, with a secretory signal. Remember that you can engineer that. You can put the spike protein. It will have a secretory signal at the front. And what will happen that if you put a secretory signal in there, or you can engineer in such a way that the antigen can be on the surface of these molecules. Sometimes it's difficult for a non-enveloped virus to do that, but you can engineer that in a way that you can put this spike protein on the surface of these viruses. So now what you have this recombinant adenoviral vector, and you put this in a cell. Remember that you have taken this E1 transcription enhancer out. So the cell is also a recombinant cell line, which is expressing this E1 protein in the cell. Otherwise, the virus will not replicate in there because it is essential, the E1, for its replication. So the cell is having E1. Once it allows, the virus is now replicating inside, and you get a good amount of these virus, and you can inject that to the human being. These are non-replicating adenoviruses that you can inject into the human beings because they don't have that E1 proteins. So the second way is the same way that what you do, that this is basically a transgene cassette that is just produced in this process. This is a direct cloning. This is a homologous recombination versus a direct cloning, just a detail of the cloning technology. But other than that, that's not a problem. And here we talked about this, that are a suitable E1 expressing cell line, which will produce these viral vector recovery and amplification, and the identification of transgene expressing the vectors, and assessment of the virus efficacy. This is how you, either this process or this process, you make the adenovirus vaccine in this process. The last one that we are going to talk about is the mRNA vaccine, and hopefully that we will be able to squeeze this in 4 minutes. The RNA vaccines, and there are many different ways it is being done. So one is that you clone the sequence into a DNA, and the DNA do contain a replicase. This is your RNA-dependent RNA polymerase, and the mRNA that will produce probably the spike protein for the SARS-CoV-2. And you put this DNA, inject that DNA. So now you have the replicase. And remember that the replicase is the RNA-dependent RNA polymerase. And then you have that transgene. So what's going to happen, that this is your positive sense RNA. You can easily see that. And this is, after replication, will produce the negative sense RNA, and this will act as a template. And you produce these mRNA transgene, which will be translated by the ribosome to produce the vaccine antigen. So again, remember that how this is going. This is happening in the nucleus. You are injecting that into the cell. It will go into the nucleus, and then the positive sense RNAs are produced out of this. So this is basically the process of the with the DNA plasmid. On the other hand, what you can do that you can package the virus-like particle, and this is a self-amplifying RNA. So the self-amplifying RNA is packaged into a virus-like particle, and this does contain the replicase and the transgene. And the replicase, this RNA-dependent RNA polymerase, is going to replicate that, which will produce the mRNA, and that can be translated into the vaccine. So this also CGMP, remember that in a clinical good manufacturing practice is CGMP. So this is also undergoing some of the trials. So the next one is an in vitro transcribed saRNA, and these technologies are probably used by the Pfizer and the Moderna, but they will not exactly tell that what they are doing. So in this in vitro transcribed mRNA, you take a replicase and a transgene, and you package them in a nanoparticle. And this nanoparticle is usually a liposome, artificial liposome, and you inject this, and you get this positive sense RNA already in vitro transcribed. This is your replicase, this is your transgene. From here, the replicase will produce, because it's an RNA-dependent RNA polymerase, remember that, the cell doesn't have that. This replicase, we can negative sense RNA and the self-amplifying saRNA. So that is the advantage of that. So now you have the template. On this negative sense, these replicase will make these mRNA transgenes, and these mRNAs can be translated by the ribosome that will confer the immunogenicity. The same way, there are two other ways, and these are even better, but the same mechanism, that here the nucleotide region is flanked by a trans-amplifying RNA or tRNA. So the difference is that here you can see that the replicase and the transgene are in the cis. They are just in a same construct. You have just one RNA in this package. Whereas here in these two, there are two individual RNAs, and they are in trans in that condition. And you have a self-amplifying replicase. This can also make the mRNAs the same way, where in the optimized tRNA, this is a non-self-amplifying replicase, and it has been seen that a non-self-replicating replicase produce even a more robust production of the mRNAs. Because that's what we believe, the just last thing is, if it is a non-self-replicating replicase, probably the cellular energy and the ingredients are not channelized towards this. All are channelized towards these mRNA and the production of this. And that may, because here you have to produce this as well as this, two products. Here you are just producing this one product, and maybe the amount is much more, 10 to 100 times much as transgene expression than this one. And these are the mRNA vaccine technologies that we have. All right, 1 minute over, but we finished it.